yeah, thank you for Erica. Thank you to Erica for inviting me. It'll be fun to chat about this. And I'm approaching this presentation with the awareness that a lot of people probably aren't neuroscientists. And so I'll try to break it down as easy as possible to give you a sense of um, what the current state of research and findings are on brain imaging studies related to the acute psychedelic experience. So that is while you're under the effects of the drug. It's developed a lot over the last 10, 11 years. And yeah, here's a little update on where things are at and what the limitations are and how I feel like things should or will be moving forward. Cool, so a little overview. I'm just gonna talk first about some very basic things around what neuroimaging is and how to understand it. Well, then I'm going to talk about some of the latest findings, walking through different categories, mainly corresponding to LSD, psilocybin, and DMT. Then I'm going to highlight some of the limitations and then some of the future directions. All right, so neuroimaging basics. So what is functional neuroimaging? It's using technology to quantitatively measure and assess the function of the living brain. Right? And it's functional neuroimaging. If we were just to call it neuroimaging, that would be like going and getting a normal MRI done that looks like your brain structure that doctors might use to try to look at damage to your brain or whether you have a tumor or something like this. But functional neuroimaging is different in that rather than measuring brain structure, it's able to measure brain activity through an indirect measure that's based on changes in blood oxygenation. But I won't go into that detail, but it's a way of measuring the function of the brain while somebody's alive and doing something or tripping on a psychedelic drug. And there are multiple technologies that have different pros and cons and are used in different contexts. So I'm going to be mainly talking about functional magnetic resonance imaging, which I just was referring to when I talked about blood oxygenation changes. And this is research I conduct. And so this is an fMRI machine. It's a giant magnet that you put your head in, and it's able to measure your brain activity. And the strength of fMRI is that it's able to measure activity while also being very, relatively precise in terms of what brain regions are activating. Contrast, there's EEG, where you have to wear a headset of electrodes, and it measures electrical activity that's detectable at your scalp. This is really good for knowing what happens at the level of milliseconds. So it's really precise over time, but it's very hard to know where precisely the signal is coming from in the brain. So as fMRI is really good spatially, locating where in space these regions are, it's not the best in terms of time, usually anywhere from 500 milliseconds at the most fastest to around 2 seconds, whereas EEG, you can get the like single millisecond range. MEG is similar to EEG, but it uses, it measures magnetic fields of neurons and is able to, it's kind of an in-between between fMRI and EEG. You're able to localize regions, which regions are active better than EEG, but not as good as fMRI, but it has a fast ability to measure the brain at fast intervals at the level of milliseconds that fMRI can't. So there are trade-offs between these three. And then a fourth one is PET, positron emission tomography. What's cool about PET is that with this technology, if you can inject radioactive tracers, so radioactive compounds, you inject a very small amount of it and it goes, and you can design them to be selective for certain receptors. So you can be like, only go to the serotonin 2A receptor, which is a receptor important for psychedelics. And then you're able to see where those receptors are located in the brain, for example. And so PET is useful because you can measure chemicals and neurotransmitters in the brain, which you can't do with these other ones. So neuroimaging in sum is using these different technologies in different ways to measure the activity of the brain. And very broadly, there are a few different types of neuroimaging studies. So there's acute, which acute means within the actual experience. So you can be used to look at the neural correlates of psychedelic experience. And post-acute is like post, so after the experience is over, how has your brain function changed? And then there's also so-called resting state versus task-based. Here's a cute image I got ChatGPT to make for me. And so a resting state is basically in the context of psychedelic research, like give somebody some psilocybin or LSD, and then just ask them to close their eyes and lie down the scanner and do nothing. And it's just like when your mind's free flowing, what happens in your brain. And then alternative to that are tasks where, because when you're in the scanner, if you look up, you usually have a screen projected in front of you. And you might have a button box in your hand, so you're able to complete certain tasks or answer questions. And then we can measure your brain and how it responds to, for example, completing a creativity task when you're under a psychedelic versus not. And so that's different than the so-called resting state where you're just sitting there and doing nothing. So the majority of psychedelic brain imaging studies so far have been using resting state. So, give somebody a psychedelic, either orally or intravenously, and just have them lie there and just trip while we're measuring their brain, essentially.
I'm David Bronner, Cosmic Engagement Officer, Dr. Bronner's board member of MAPS. Subscribe to MAPS Canada. Bam!
And so one thing helpful here is to somebody needs to be muted. Something helpful here is knowing what level of the brain we're talking about with neuroimaging, right? So we can start with the very smallest unit, which is a neuron, right? And then the receptor that psychedelics target to elicit their main effects is the serotonin 2A receptor. I won't be going into that in this lecture, but most of you know that, I assume. And that's located at the neuronal level, right? It would be on the dendrites of this neuron, which are these protrusions right here where my cursor is. Hopefully you can see my cursor. And then these neurons group into populations of what's really like a few 100,000 neurons, and then hundreds of thousands of neurons. These populations group into brain regions, which there's hundreds of in the brain, depending on how you size it up. And then these sets of regions combine to form what are called large-scale brain networks. And so usually fMRI research or neuroimaging research is looking at the level of brain regions and networks. And so this concept of a network is really important, and a lot of you have probably heard about the default mode network or other networks. And so what is a network exactly? As I kind of just said, it's a set of regions that communicate with each other more than they communicate with the rest of the brain, right? In other words, they preferentially interact. And usually it implies that these set of regions share more information with each other to perform a shared function. And each network has a distinctive set of functions. And commonly we view the brain in terms of 7 networks, but there's many different ways to decompose it, to parse it up. And this 7 network parcellation is shown here. This paper has been cited thousands of times, and people use this very often. Each color is a different network. So very quickly, some of them. So in the purple, in the occipital lobe, is our visual network. It receives information from our eyes, and it mediates our visual perception, right? And then this light blue one is our somatomotor and auditory networks. So our sense of touch, our ability to move, and our ability to hear things often gets grouped in the same network. And then in the magenta-ish color is everyone's favorite network, the default mode network, which is a distributed set of regions along the middle here, but then also on the side, on the lateral surface. And so a little bit more about the default mode network. It's interesting because it's the most expanded part of our brain in humans relative to our closest cousins, which would be chimpanzees and bonobos. And so it's what arguably mediates what makes humans distinctive as a species. And in other words, it's involved in our ability to create abstractions and think of things that are not in the here and now. And it's involved in anything essentially that's based on memory. So when we're thinking about ourselves and lost in our own personal narrative, default mode network. We're watching a movie, we're trying to interpret what's happening, default mode network. You're trying to imagine a future experience or remember a past one, default mode network, right? It's involved in a lot of processes that rely on memory. And again, allow us to abstract away from the here and now into some imagined reality. And critical for our sense of self, for our sense of who we believe we are, who we've been, who we're going to be, etc. Often people refer to that as the ego. That's a different terminology, similar but different depending on what system you want to link that word to, but broadly that's what people refer to. And something interesting is that this part of the brain has a very high density of serotonin 2A receptors. And so this is the receptors that psychedelic mainly target to elicit their effects. And so you see in this image, red is the high density of serotonin 2A receptors. This is based on PET imaging, which I mentioned earlier. And you can see this overlaps with the magenta in this network image. And so based on where these receptors are located, we would just assume that psychedelics would be affecting this network pretty strongly. Okay, so now you know what networks are, you know what neuroimaging is, so let's talk about some of the findings. First, here's a really great image. I'm not going to go through this in detail, but just to give you a sense of the landscape, this is from a review paper by Drummond McCulloch, who's like a collaborator of mine. He's at the University of Copenhagen. This was in 2022, so it's not completely up to date, but like gives you a sense of the number of studies that have been published to date. Each green study is with psilocybin. This bluish maroon color is ayahuasca, and then blue is LSD. And this is how many data sets have been collected so far. And you can see something I want to draw your attention to is where it says participants, the number of people in each study. It's like 15, 15, 20, 22, 23, 15, 10, 21. You know, roughly an average of something like 15 to 20, which is very, very small, right? Just so you know, like these studies are really hard to conduct, they're expensive. These data sets are small, and like ultimately each one is preliminary. But together we can start to try to draw some conclusions. Yeah, that's all I want to say about this one. This is the landscape so far. And okay, so let's move through some of the main findings. So first, let's talk about brainwaves. So MEG and EEG, because they can measure at such a time-sensitive resolution, you can measure brain waves. So how do neurons oscillate, so activate, turn on and off in synchrony with each other in different frequency bands, right? So oscillatory power is basically the strength of a brain rhythm, corresponds to how many neurons are synchronizing in that frequency, right? So like higher frequency power or oscillatory power means more neurons firing in synchrony at that frequency. And the thing with psilocybin and LSD is that they reduce the synchrony of neurons across all frequency bands. So we call this broadband desynchronization. So this means they're perturbing the ability for neurons to fire in these coordinated synchronous ways. And this is strongest in the alpha band, which I'll talk about in a moment here. So this image here is with psilocybin. This is with LSD, known as only blue, which means reductions. And what's interesting is reductions in the alpha frequency band, so less alpha brainwaves, has been associated to more visual effects, so seeing images and complex scenes, and also more ego dissolution, depending on what part of the brain it happens in. Something interesting about DMT is that you also get reductions in this alpha, but you get increases in delta, which is often found in closer to sleep-related states and deep relaxation. And then gamma, which is interestingly involved in perception and like very fast processing and binding of information. So you get slightly different effects with DMT in terms of brainwaves, but the commonality again is this reduction in alpha, which likely strongly relates to visual type effects with DMT as well. And a last thing you can measure with brainwaves is like how diverse or how unpredictable is the signal over time. And this is found to be increased with all three drugs. Here's an image from DMT. If you look on the right here, Steve on the left, DMT on the right, you can see there's a significant increase in signal diversity, which means the brain waves or the activity is much more unpredictable and entropic. So this is at the level of brain regions, right? So the takeaway here is that most frequency bands get desynchronized. So it's a kind of disruption of usual processing. Then if you scale up from that to networks, it has changes which span the entire brain, right? So within networks, so how much a network's integrated with itself is reduced for most networks. And so this image is from LSD. And so for each of these images, you'll see 0 and then 20, and then it goes negative. And blue is how integrated this network is. So for visual, for default mode network here, and for several others, it's all decreases in that integration. So these networks are becoming less integrated. So these regions are no longer as tightly knit as they usually are. And this is particularly strong for sensory networks. It's the visual network and somatomotor network and somatosensory network. as well as default network that I've talked about, and the frontoparietal network, which is our prefrontal cortex and involved in our ability to plan and reason in coordinated ways. But something I want to highlight here is that this set of findings has been found with escitalopram, so an SSRI, with salvia. Certain of these effects have been found with alcohol. And so we really don't really know what these effects mean because they happen in a variety of non-psychedelic contexts. So that's an important thing to keep in mind here. And then so I was talking about within networks, so between networks, very commonly networks become more integrated with each other. And so this image here, all you need to know is these red squares. Each red square with an asterisk means that's two networks having more connectivity with each other with psilocybin. So you can see the number of blue boxes is very limited, and it's mostly networks becoming more connected. Right, this is with psilocybin, and this one, green means more connected. You can see the majority are green, so networks are becoming more interconnected. And then this one, red, is more interconnected again. These are the ones that are significant statistically. And so this is 3 data sets. You're really just finding most networks are becoming more integrated with each other. However, when you look at the specifics of what networks are implicated in each study, they're inconsistent. And one study from Felix Muller from the University of Basel, they took 3 data sets, 2 with LSD, 1 with psilocybin, and they asked, what are the overlaps in specific networks that were changed? And so in this picture here, there's like 10 networks at the top, and red means one study, green is another, and blue is another one, right? So you can see there's not a single network change that was found for all 3 data sets. There are a couple, times where there's 2 data sets, it's green and red or blue and red, but there's never a complete overlap. And there's overall pretty poor overlap, suggesting even though we're getting this general trend, the brain is becoming more connected, the specifics is very inconsistent. There's no consistent pattern that represents the psychedelic state. And it's an open question whether that's just because these samples are small and they're limited, or like we can't expect a common state in the psychedelic state. That's a question I'll return to in a second here. You might be wondering, I came here to hear about the default mode network and ego dissolution, because this is something talked about a lot. And so let's look into some of the research here. So with psilocybin, actually, contrary to popular belief, disintegration of the default mode network as the network as a whole was not significantly correlated with ego dissolution. But studies did find correlations between ego dissolution and changes in specific regions of the default mode network, particularly those involved in memory. And so these areas, such as the hippocampus, have shown reduced connectivity with the rest of the brain, less glutamate levels, and reduced alpha power. And these have correlated with ego dissolution, but not the default mode network as a whole. So disintegration of the default mode network as a whole has not been found to correlate with ego dissolution in psilocybin. It might be a surprise for many. Here's a scatter plot where ego dissolution is on the x-axis and decreased connectivity between these 2 memory-related areas in the default mode network, the parahippocampus and retrosplenial cortex. You find that the more decreased connectivity, the higher ego dissolution, right? But you can see this is messy because a lot of people put 0 for ego dissolution in this study. Again, highlighting the limitations of these small samples because you need a lot more to get a reliable correlation. With LSD, they did find a correlation between ego dissolution and default mode network disintegration. It was actually barely statistically significant. And this was again driven by regions involved in memory and spatial location. And so it seems like memory-related processes are important here, not the default mode network as a whole necessarily. And then beyond the default mode network, disintegration of the salience network, which is involved in integrating bodily signals with our cognition, was associated with ego dissolution, as well as more connectivity between the different hemispheres of the brain. With LSD, the anterior insula and angular gyrus became more connected with the rest of the brain. And these are regions that are also broadly involved in our sense of spatial location and our perception of our body. And so in addition to the default mode network memory areas, which are probably involved in our narrative and our ability to think about ourselves, there's also regions more involved in our bodily felt and somatic experience, which are involved in ego dissolution. suggesting it is a complex phenomenon that involves body and mind, body and our narratives, with distinct regions involved in each. And so another measure that's being used is this measure called global functional connectivity. So this asks, how much does each region of the brain connect to the rest of the brain, right? And there's been several studies from Imperial College London which have looked at this with LSD, psilocybin, and DMT and found a very similar pattern in each one, where there are increases in these red areas and decreases in these blue areas, right? And so if you remember what I've been showing you, the red areas are basically the default mode network. And then the reductions are the somatosensory and motor and auditory networks and some visual as well. And so this is a pattern found in 3 data sets where the default mode network becomes more integrated with the rest of the brain, essentially. But interestingly, at the University of Zurich, applying these same analyses to their psilocybin and LSD data sets, they find a very different pattern. And what's pretty crazy about this is that it's almost the opposite pattern. If you look at the blue on this one, it overlaps with the red in this one. So it's like, okay, so there's 2 reputable research institutions doing similar research and finding the opposite pattern, which is kind of wild, right? And so actually, I don't have figures from, well, actually these are from it, but I am leading a collaboration with both Imperial College and University of Zurich to bring all this data together and analyze it, with each other in order to find out what the source of the differences are. And a little teaser on that, the paper is probably going to come out and then we're going to submit it in the next few months. But what's interesting is that so what I'm showing you here is averages, right? Averages about 20 people. And so what we're finding is that if you look at the individual subject level, there are people in this data set on the right that show this left pattern, and people in this left data set that show the pattern of this right side. And so essentially, you're getting such variability within each data set, such that 2 people in the same study might show an opposite pattern. And so what this is reflecting is this, like, how variable it is across individuals, and it's not necessarily something to do with each research site. And so moving forward, when you look at how there might be different subtypes in how people respond to a psychedelic. And this is being shown here. And this also, again, supports why we need really large samples. And so we're able to see the general trends and not just get stuck in whatever selected group of people we happen to choose. And another interesting because I read the study, is to characterize the brain in terms of this gradient that separates the default mode network from sensory areas. And this is a core organizational feature of the cortex, separating our ability for abstraction from our ability for sensory processing. And usually these are quite distinct in the brain. But what we're finding, what I found with psilocybin LSD as well as DMT, is that this is less the case. And so in this image here, in red are the sensory areas, and then blue are default mode network areas. And what this is showing is that the sensory areas are increasing in value, so they're becoming less negative and moving towards 0. And then the default mode network areas are becoming less positive and moving towards 0. So basically, both ends of this axis of concrete versus abstract processing are coming together. So abstract processing and concrete processing are less distinct from each other. So there's more of a mixing between our abstract thoughts and memories with our visual perception and our sense of touch. I won't get into this figure, it's a bit too complicated, but in general, viewing the brain as this kind of axis from concrete to abstract, and that's being reduced in the psychedelic state might be an interesting way to summarize these effects. Another interesting set of findings with psychedelics is that they make the brain more complex or diverse or entropic in its activity. As the brain is exploring a greater range of states than it usually is, This might correspond to the freedom of thought that people experience with psychedelics and the ability to think in new ways and escape any ruts they might have found themselves trapped in. And so you get increased entropy, which just means the brain signal becomes more unpredictable over time, greater number of brain states. And here's a good way to illustrate what I mean by brain states, right? So on the top, well, let's say there were four regions here, and you could say that there are different ways that they can connect with each other, right? So in this top row, they can connect this way, this way, this way, and they have a limited number of ways they can connect. But then when you take a psychedelic, there's all sorts of ways they connect, which they usually never do, right? So there's greater variety of these interconnected states, and each one corresponding to a different way of thinking, you could say, or a different mental experience. And so multiple studies, analyzing data in different ways, have found that these states become more diverse in the psychedelic state. And also in this really interesting study, it actually suggested, I won't get into the technical details here, but it suggested that literally the amount of energy the brain needs to use to switch between brain states has been reduced. So it's like, greasing the wheels of the brain, and it's able to slip into different states much more easily when using less energy. And this is what, again, might facilitate a kind of flexibility of mind that's useful for therapy. However, I want to highlight here, there are limitations in that there are many different ways to measure entropy and complexity in the brain. And some of these give results which are not really consistent with each other. They're a bit different, and it's hard to know what exactly each different measure contributes and what it means. And it's unclear what the most consistent finding is here. But generally, broadly speaking, evidence suggests that the brain is able to be, activate in more complex ways dynamically and enter into new states. But there's still a lot unresolved there. And another region that's talked about a lot in psychedelics is the thalamus. So this is the region of the brain that is kind of like a relay station for sensory information. And so information from our eyes, from our ears, goes first to the thalamus, then to the rest of the brain. And so it's able to control what information reaches our consciousness, essentially. And some studies have found that the thalamus becomes more interconnected with the rest of the brain in the psychedelic state. You can see in this picture on here, placebo is on the top, red is where the thalamus is connecting to, and then with LSD, and then the differences. And so you can see there's much more connectivity between the thalamus and much of the brain with LSD. This is also shown in this picture. Where these lines are originating are the thalamus, and each one that is red and also green, they indicate increased connectivity. So this suggests that the thalamus is now sending much more sensory information to the rest of the brain. It's not filtering as much as it used to be. This is what it suggests. And some other studies have found that this increase in the connectivity of the thalamus might be highest for sensory areas, which makes sense based on what I just said, because it's now not filtering the information it usually was. And so now it's connecting and showing much more information with areas related to our sense of touch and our vision. But the thing with the thalamus is that it has different subdivisions, which do very different things and have very different connectivity profiles. And so these studies I'm showing on here on the right just took the thalamus as if it was all one thing and then correlated it with the rest of the brain. But like that's averaging over very, very distinct places. And so our recent study looked at specific regions within the thalamus and actually separated the thalamus into 8 different regions. And then when they looked at that fine-grained level, they actually found reductions in the connectivity between the thalamus, which are these blue lines here, and most other networks. So it could just be those increases we're seeing were just because you're blurring across all sorts of things and getting some average that doesn't really exist in any individual part of the thalamus, if that makes sense. And so again, things are complicated here. It's unclear whether we can view it in the way that people usually do, that it's reducing the gating of sensory information and flooding the cortex with sensory information. It seems to be a bit more nuanced than that, and we need to look into more detail at different parts of the thalamus. And so there is some evidence that the thalamocortical theory, which I just mentioned, of sensory gating and filtering, might be true to some extent, but more research is needed on how different parts of the thalamus do different things. Another interesting region which has been flagged in psychedelics, in the psychedelic world, is the claustrum. And this region has a high density of serotonin 2A receptors. And something this region does is it controls the different networks in the cortex. And so in this image, it's showing, in red is placebo, and then in this pinkish color is psilocybin. There are several ways that the connectivity between this region and the cortex is changing, including in relation to the default mode network. And so claustrum is kind of this region that's behind the scenes and kind of pulling the puppet strings of different networks in the cortex. And actually, this is a really messy scatter plot, but there is a correlation here somewhat between how much the claustrum connects to the default mode network and its connectivity. And so in the psilocybin state, the claustrum is less connected to the default mode network, and this is associated with default mode network disintegration. And so it could be part of the effects we see at the level of networks are driven by this other region called the claustrum. And so that's emerging as another important area. Okay, so that was a lot of stuff. What are the takeaways here? So in general, networks become less integrated within themselves and more integrated with each other, potentially suggesting a blurring of distinct functions in the brain and a reduction in the ability for things to happen in a specialized, separate way. But as I highlighted, the specifics of which networks are being altered and which way varies a lot across studies and also very likely across individuals, even to the extent that some studies are finding opposite findings, as I mentioned earlier. And so this, there's a lot of complexity here, and it's hard just to average across everyone and try to find a single pattern. In fact, I've argued in a paper that that's probably not the way to go. We embrace how everyone responds to a psychedelic differently, and that's going to show up in their brain as a different pattern. But more on that in a second. And if you don't look at networks, but instead look at how abstract versus concrete processing is changed, you see that then there's a consistency across these different data sets. And so this more general way of looking at it in terms of a differentiation of abstract versus concrete processing might be a more accurate, or not more accurate, more useful and way of looking at it that can help coordinate it across studies. And then there are limitations with this as well, but the evidence does suggest that the brain's dynamics, its activity over time, and how brain regions interact become more complex and entropic, meaning unpredictable, with a greater range of states. Finally, a lot of these effects I just mentioned might be modulated or manipulated by these kind of subcortical older areas of the brain called the thalamus and claustrum, which actually manipulate the networks of the cortex. And so it's a complex set of things at play here, modulating what we see in the brain. But as I highlighted, details are unclear and we need more studies. So given all those findings, there are some additional limitations to keep in mind on why, a lot of neuroscientists are much more hesitant around making strong claims than, the news media and a lot of people are, right? So one thing I flagged a few times here is that these studies are with 15 to 30 people, right? And we know at the very, very minimum, we need at least 50 to 100 to get some semblance of reliable effects and also to find correlations with experience. Because here's why, right? Let's say there are 100 people and, about 30, one third of them respond one way, another third respond another third respond another. If you're just going to take 30 people, you might get a disproportionate amount in that first way of responding. Then in another study, you take another 30 people, you get a different overlapping set of people. And then those two studies are going to be inconsistent, right? Because you haven't fully sampled the variability that's out there. And so this is why in studies moving forward, we need much larger samples. So we're able to actually get a more even distribution of the different types of people, and then really look at what's common across them and what the different subtypes are. So this is why these small studies are hard, because you might just get locked into a non-representative sample. And this is what we refer to as sampling variability, right? As you take different samples, these distinct samples, if they're not big enough, are going to vary. And that's like that's like probably the main problem in psychedelic neuroimaging is that. And related to this, we need to look at how the effects vary across people, right? Rather than just trying to find some average thing, this is your brain on psychedelics. I think that's kind of ridiculous because if you think about it, each person's psychedelic experience is so different, right? And even for a given person, the different experiences you have are going to be radically different. So then how can we expect there to be one brain pattern that represents a given individual all the time, and then and then if you can't expect it at the individual, how can you expect it across 20 people, right? And so we need to be looking at how are people individually responding to psychedelics? What are ways we can predict that? What are different subgroups of ways that people respond? I think that's much more meaningful than trying to find the pattern that represents the psychedelic state, as if there was one psychedelic state anyway. And some other limitations here. These brain imaging studies or technologies are very sensitive to people moving their head because your brain is being scanned in this very precise way. Even if you move like a millimeter, it'll affect the data. And so when people take psychedelic drugs, it's harder to sit still in the scanner. And so people's head motion is a major issue that we have to tackle as neuroimaging researchers. And in response to that, there's a lot of ways to try to get rid of using statistical techniques to get rid of that head motion, and that varies across research labs, sometimes with effects on the brain data that are hard to predict. And then not only that, it's really hard to compare across studies and research groups because people do different statistical approaches, different statistical analyses. And so you have slightly different analyses in two different places. It's unclear whether what you're seeing is a difference in the data or a difference due to just the statistics that you did. And so there really needs to be more coordination to make good comparisons across studies. And this is an issue across all of neuroimaging. Also, studies vary in the dosage, right? Is it 75 micrograms of LSD? Is it 100? Is it 150? And then also route of administration, is it given intravenous, injected, or are you taking it orally beforehand? So that's going to change when the peak of your experiences and maybe how intense it is and a variety of other factors that might affect the brain effects. And so these are different across studies as well, making it hard to compare a lot of the time as well. Okay, so all that said, what are some ways forward? So one thing that I proposed in a paper earlier this year is that rather than trying to find some canonical pattern of the psychedelic state, recognize that what psychedelics do is they change the way that the brain functions. And that change in functioning is going to play out for each person depending on their unique set and their setting. And so there's like a general way that the brain's going to process information differently, and how that manifests is person specific, right? And so the general way that it changes the brain is that the brain becomes more flexible, more dynamic, more diverse, more integrated across the entire brain, different networks you're communicating. And as a result of all that, it's tuned for information sharing. Information is being able to move throughout the network, throughout the brain in a more efficient, easeful way. And this is associated with the concepts of criticality and metastability from complex systems theory. I'm debating whether to go into details in these, but basically the gist here again is that the brain is at this place where under psychedelic, it gets pushed towards having more complexity, a greater number of ways of responding to information, and a greater sensitivity to information. And then this, again, will play out in different ways depending on the person. Here's it visualized a bit. So if you look at this top row of brains, if you look at SO1 here, this is baseline. Your brain might slightly change, it doesn't change that much. It's the same general pattern if you look across these different subjects, right? Take a psychedelic, and now for each interval of time, brain is entering in a totally different composition, right? Because the brain under a psychedelic is so complex. You're jumping from here to there, here to there. There's a lot of experiences you can have within a short period, and that's represented in the brain, right? And then if you want to think of it, you're exploring this abstract mental space of possible ways of thinking. At baseline, you explore this kind of like general, pretty small range. You have generally the same kind of thoughts and experiences throughout the day. Then when the psychedelic happens, you're blown wide open and you're able to explore these different trajectories of mental states and ways of thinking. And again, how that plays out for the individual is going to be different, as shown by the different colors. These different squiggly lines are going to explore, like they all share the property of being more flexible, but where they explore and how they change is going to be different. Something else I'm helping lead with a couple of researchers is a coordinated effort across every psychedelic neuroimaging lab in the world to bring together all their data and pre-process it, denoise it, and analyze it, and all together in one analysis to find what's common across all these different data sets, and set a benchmark, like the groundwork for studies moving forward. So we've been meeting for over a year for this. There's research institutions in the United States, in Switzerland, in the Netherlands, in Denmark, in Brazil, in a whole bunch of places internationally that have joined this, and we have shared all the data sets, total of 11 data sets, and we're analyzing them all together. So that's to come as well. Moving forward, what we need more of are coming up with specific hypotheses and then saying what we're going to do ahead of time, pre-registering it, and then conducting that study. We've done a lot of studies like that have basically just been, let's give people a bunch of psychedelics and look at their brain. But I think now we're at the point where we can ask specific questions. Let's give a psychedelic and test whether specific parts of the thalamus are connected to these specific parts of the cortex. And then we know ahead of time, this is what we aim to look at, and it's more statistically robust and reliable, like to look at that specific thing and do what we say we'll do. So I think we need more hypothesis-driven studies. And just larger samples, as I'm sure you know why at this point, and greater coordination across research labs in approaches to denoising and analyzing to help comparisons. And looking more, I've already said this, at individual variability and how to predict that, as opposed to just looking at group level averages. And using tasks or having them watch a movie or nature scenes or complete something in the scanner other than just sitting there in an uncontrolled way, tripping, which is limited in what it can tell us a lot of the time. It's very useful to probe the brain by getting someone to do something and then measuring how that's different under psychedelic. Lastly, what's really important is coordination across labs, as I kind of mentioned before, but specifically in terms of the analysis. Cool. That was a lot, but that's everything I got. And happy to answer any questions now. And follow me on social media and YouTube if you want some more psychedelic science videos.
Thanks so much for presenting today, Manesh. That was an excellent presentation. It's great to hear about the advances that are being made right now to standardize and push forward psychedelic neuroscience.
Sure, my pleasure.
Yeah. So I'll go into some of the chat questions. So Julie's first question, when you say eco-dissolution, are you referring to the subjective experience during the trip?
Yes. When I say ego-dissolution, I'm referring to people in the experience feeling like their sense of being a distinct individual with a certain narrative and identity is kind of dissolving or going away. And these are things they report on using questionnaires.
And then this next question, so could maybe differences between individuals be attributed to the intention of the participant?
I think definitely. I think set and setting, right? And a lot of set is just who you are intrinsically as a person, right? Like your set is like mindset, so the psychological factors that influence the experience. Each person is different. We have a different life experience, different personality, different sensitivity to the drug, all these things, and different brain, architecture, brain connectivity. And all these things are going to play out in your experience, and you're going to go into a different type of experience. And intention is, of course, a major influencer of our experience, and therefore it's going to influence how the brain responds.
And then another question is, do any of these findings apply to ketamine?
So not all of them. Ketamine is different in how it affects the brain, but one way it overlaps is that I mentioned earlier that there's some research suggesting that the default mode network and including areas of our prefrontal cortex become more connected with the rest of the brain. And with ketamine, pretty reliable finding is that your prefrontal cortex becomes more connected with the rest of the brain, and that's related to some of the antidepressant effects of ketamine. And so this increased connectivity of prefrontal cortex areas is common across the different drugs.
And then I'm just going through as Laura sent quite a few questions. Could you talk about like the questionnaire that's given to a person after the trip to gather information about their own experience?
Yeah, so there's several ways to do this. The most commonly used one is the 5D ASC, as it's called, the five-dimensional altered states of consciousness questionnaire. And this is like super long 94 question questionnaire that looks at different aspects of the experience, right? It looks at ego dissolution type effects, changes in the meaning you attribute to things, simple geometric shapes that you're seeing, or like complex images of scenes and landscapes. It also measures anxiety and other like negative aspects of the experience. And so we get this like quantitative, comprehensive look at the nature of their experience. And yeah, so that measure would then be correlated with different aspects of the brain.
And I was just curious, like, so the MEQ30, is that like a shortened version or an entirely different scale?
So it's an entirely different one. So like the one I mentioned, so the thing with the 5D ASC is that it's often to some extent used to correlate with the brain, whereas the MEQ was designed to correlate with changes in depression and changes in well-being. So slightly different, but like, yeah, it's a similar concept of a measure aimed to look at the brain, or sorry, the experience when you're undergoing it.
Yeah, that's really interesting to hear the difference. Next question. So you said that the thalamus needs more research to understand what subdivision does. So would finding correlations between perceptions, experiences, and maybe what shows on the imagery help with guiding this research?
Well, the interesting thing about the thalamus is that some parts of it are associated with visual perception. Some parts of it are associated with our sense of touch. Some parts of it are associated with types of abstract thinking, and then our hearing. And so literally it's separated based on type of sensory processing or mental processing. So I guess what you're alluding to here is true, that if the thalamus is separated in that way, which it is, then maybe looking at correlations with auditory versus just visual versus just mental, and then doing that in relation to specific parts of the thalamus is the way to go. And I would totally agree. I think that's what we need. That's what I was kind of referring to when I was saying pre-registered hypothesis-driven studies, right? It's like based on what we know about the thalamus, based on what we know about psychedelic effects, I hypothesize that this effect will show up here, etc. And so, yeah, totally.
Right. And then next question from Laura. So in regards to separating different types of experiences, so having a solid questionnaire could help with high individual variability by being able to group similar experiences. So maybe expanding on the psychometric scales that are currently used to measure psychedelic experiences, like, is there any way to categorize them in a way?
Yeah. I mean, this is definitely it's tricky. Yeah, it might be possible with existing research to try to like cluster together groups of people who respond in similar ways across the different measures. Yeah, I wonder if that's been attempted before. The main limitation is you need a lot of people to do this because you want enough of each different type of responder, right? And some of the work I mentioned that I'm doing on the brain level is basically looking at that. It's like taking two distinct data sets of people who took LSD and was brain scanned, and then pretend that it's all one data set, and then in kind of like try to cluster it based on the data alone, which subjects are more similar to each other, right? And then I did that actually, and what it showed is that like 30% from one and 70% from another are the same, and then vice versa. And so it didn't just tell me like, oh, these two data sets are different. It's like they're overlapping because there's subjects that are similar in each. And so I think that's the way to move forward, because you can do that with questionnaires as well. You just need hundreds of people to do that properly.
Yeah, that would be a really interesting way to go forward and see maybe maybe we can predict how people will respond to the same dose going forward, of course, probably further in the future. And last question from Laura. So what is the main bottleneck to finding more people to participate in studies?
Cost. It's pretty expensive. Like scanner time at an institution is like $500 to $600 an hour. And then if you bring somebody in to do psilocybin, you usually have to have a physician there, for like 6 to 8 hours. And doctors are expensive, right? And so it's like 6 to 8 hours for each person. So yeah, things add up really fast with psychedelic research. But the main issue there is that a lot of this research is done through philanthropic donation, right? The amount of federal funding in Canada or in the States is still relatively low, although it's starting to change. We're hoping to see more grants coming out. But for example, I work here with Robin Carhart-Harris, who's one of the leaders in the field. He's mostly working off of philanthropic money, not from government agencies, and he's like the top researcher who should be getting money from federal sources, right? But hopefully that is changing. We're applying a bunch of things together, and he has lots of stuff on the go, of course. But so far, it's been like rich people giving us money, basically.
The biggest issue of psychedelic research, held back by funding.
For now.
Nikita wonders, how do you measure entropy?
So there are many different ways to measure it. So one popular one, and this is going to sound abstract, but you asked for it. It's like if you imagine okay. So let's say you have 10 minutes of a brain recording, right? And you have the activity every minute. Let's do every second. And then what you would ask is, and let's say the signal can be 0 or 1, right? So like for 10 minutes, there's a bunch of 0, 0, 1, 1, 1, 0, 1, 0, 0, right? And then the question is, how many different patterns is needed to characterize the data? So let's say if it was all zeros, then all you need to know is 0. But if it was 0, 1, 0, 1, 0, 1, 0, 1, then it would be 0, 1 or 0 and 1. There are 3 patterns there. Does this make sense? But let's say it goes like 0, 1, 1, 0, 1, 1, 1, 0, 0, 1, 2, 1. Then you need a whole bunch of different mini patterns to be able to characterize that. You need 3 1s in a row, you need 0, 1, 0, you need 0, 1, you need 0, 1, 1, 0, right? I hope this makes sense. So there are many different sub kind of strings or sub patterns you would need to recreate the entire thing. And so one way of measuring entropy is how many different sub patterns are there in the signal. And if there's more, that means it's more unpredictable because you never know which pattern is coming next. And so entropy is really just like one way to think about it is like, if you were to try to compress that data into a more simpler form, how complex would that compression be? Like how many different components, how many building blocks do you need to recreate it? So that's one way Lempel-Ziv complexity is computed.
Yeah, no, thanks for going through that. Like, I feel like entropy is like a tricky one to really grasp, and I think you put it very well. So Nikita also asks, does the increased thalamic functional connectivity that you found last over time? Because many people report that they're more sensitive to stimuli after psychedelic treatments, which could be related.
Yeah, it's a good question. I don't think that's been specifically looked at. That would be an interesting one. Because I do think, there is the afterglow phenomenon, right? Where the next day you're a bit more open, things might seem a bit more vibrant and lit up. And so I would guess there very likely is, or very possibly is, an increase in thalamic functional connectivity to visual and sensory areas that lasts at least 24 hours. And this is something we have data to test, actually. So that is an interesting question. Yeah. And then it would depend, as I was saying before, on which areas of the thalamus, right? But like, it is plausible that that happens, but hasn't been looked at yet, as far as I know.
A bit of a fast question. So what sort of physical symptoms do you track in studies, such as vibrations, heats, or cold sensations?
Interestingly, not those. Usually when we look at physiological signals, it's like heartbeat, body temperature, or I guess heat is one of them that you mentioned. blood pressure, heart rate, body temperature, stuff like this, these physiological metrics, which are used to measure safety basically by the doctor. It's like your vitals basically. Um, but the studies we're doing here at UCSF, we're also doing, we're doing respiration, we're doing muscle changes in the face. We do electrocardiogram, so heartbeat things before and after. And so there are more and more physical things being tracked, but things like vibrating or like temporary sensations throughout the body, like that's not necessarily examined yet.
Yeah, and I guess when I read the question, I first thought like just the condition of the like the lab and the scanner, but I suppose like as long as it's decently standardized, that's not an issue. Interesting question. Julie would like to hear more about the link between the default mode network and eco-dissolution, or the lack of.
Yeah, so the takeaway from what I said earlier is that often, with Michael Pollan and the whole thing, it's like people associate default mode disintegration with ego dissolution. But as I was saying, with psilocybin, didn't even find that. With LSD, they kind of did. But it seems to be more nuanced, where it's not the entire default mode network, it's specific parts of it that are important. And also regions that are independent of the default mode network entirely. So within the default mode network, it seems like memory regions such as the hippocampus and parahippocampus and their connectivity with the rest of the brain, and when that's altered or decreased, then people have more eco-dissolution type effects. And that might correspond to a cutting off of our autobiographical memory, right? A cutting off of remembering who we are and contextualizing our experiences in terms of this sense of self. That might be disrupted, and that's related to these memory areas. But then in addition, outside of the default mode network, there are areas such as the insula, which really are connected to our spinal cord and integrate feelings and sensations from our body with our brain and our thinking, and which correspond to our sense of embodiment. That also is disrupted, and that also relates to eco-dissolution. So there are different components, some of which might be related to DMN, and some which aren't, which are both involved. And basically, we need more studies to look at this in more detail.
Ali is interested in, over the course of a psychedelic experience, measuring with functional imaging at multiple time points, what the results of those studies have been.
Right. This is a great question because we're running this study right now. So at UCSF, where I am currently, we're doing a study where we're taking people and putting them through 4 high-dose sessions of psilocybin. 10 milligrams and then 25 milligrams, which is like a gram to 2.5 to 3 grams afterwards. We're doing that every week for 4 weeks for 10 different people and looking at, how does their brain respond differently across multiple consecutive sessions using fMRI and EEG. And then how is that within subjects, and then how is that different across subjects? So we are running that. We don't have any results, but I can't say any results, but we're on our 2nd subject out of 10 doing that. And we're basically, that's all of next year, we're going to be running this study. And so then following year, 2025, it'll probably come out. This research takes a long time.
And Rael wants to know, so you found some of these effects are same as found with other substances such as alcohol. So are there specific differences that you found with psychedelics?
Yeah, interestingly, a lot of the stuff, I think, and this is what I argued in my paper, the complex systems paper, is that what matters more is the overall whole brain pattern rather than specific networks or regions, right? It's like, but even more than that, it's like, okay, if we were to ask, is different brain networks becoming more connected to each other and less connected with themselves? Is that specific to psychedelics, right? It's like, no, actually, as you age, that happens naturally, right? And it's not that 60-year-olds are just psychedelically tripping all the time. It's that their brain is less able to separate different networks as it gets older, right? But psychedelics obviously do that similarly, but like with very, very different effects. So there's something different there. And so one difference was in the whole gradient thing I mentioned, which was separating this access from abstract default mode network to sensory processing, visual network, somatosensory, like making that axis like collapsed seems to be specific to psychedelics. So that's an interesting signature that's not found with aging, and to my knowledge, not found with other drugs. And so, yeah, so we're in the process of trying to identify what these different specific things are. Another one might be the way that the brain enters into a much more variable or wide range of different states. That might be, and the whole entropic thing might be somewhat specific to psychedelics. Although there was a study with caffeine that found increased entropy in a bunch of the brain, which again, it's like super complex because it actually relates to what specific measure of entropy they use for that, and it gets really messy. And so yeah, I mean, things are much more complex than people usually realize, and there's a lot of caveats, and we're super early with all this stuff, so it's really hard to say.
I'm interested if there's maybe any other areas that you would look at in terms of like finding similarities and differences. Like, for example, the parallels people always try to draw between psychedelics and schizophrenia.
Yeah. I mean, this has been looked at in different ways. There are some overlaps and some differences. It's interesting. I mentioned the gradient collapsing thing that actually was found with schizophrenia to some extent as well, although with slight differences. And so, yeah, there is some overlap there, but also some big differences. But it's hard to compare them because schizophrenia is like this chronic condition, and a psychedelic experience is this 4 to 6 hour thing, right? So unless you be able to get people in as soon as they have their first ever psychotic episode, throw them in the brain scanner. But that's you can't really do that anyway. They're not going to be the best subject, right? So it gets very hard to compare. But on the drug side, people are comparing things like psilocybin and LSD to MDMA, to cannabis, to things like salvia, and then also amphetamines, just to see, you know, what's very specific to it. And so there is stuff aimed at that as well.
Yeah, and that's fascinating to see. Like, I was just like thinking about this today, like how much have we figured out about other drugs when people, you know, point to like such a big mystery with psychedelics. I feel like in all aspects of research, there's just going to be like big gaps, especially in terms of the method issues that you brought up with like the lack of standardization for different methods of statistical analysis and adjustments. You know, would you say that's something that is like very prevalent in like neuroimaging generally, or is there something that lies in psychedelics that makes it challenging?
Right. I think both. I think it is present throughout the field. Is this kind of like, yeah, inconsistencies in approaches and so on. I think like given that psychedelics are so complex and so variable across people, it's just like amplified or highlighted with psychedelics, but it is present in different ways with like most drugs.
All right. Thank you so much for answering our questions. Actually, we do have one quick one. Vidya is just curious about aneurysms and whether those are a common exclusion factor for psychedelic studies.
I mean, I'm not a neurologist or a psychiatrist, so I can't give medical advice on this. I would recommend asking your neurologist or whoever you've been going to for your aneurysm. Yeah, it's hard to say. It depends on your specific case. Although there's a lot of evidence for using psychedelics for things like stroke and traumatic brain injury and these kinds of stuff because it's anti-inflammatory and so on. But yeah, I can't give any specific recommendations here. You'd have to ask a doctor.
All right. Does anyone have any like final questions? I feel like we put Manesh through a pretty comprehensive Q&A session. So we can end off. What are you most excited about for the future of this field and research?
Sure. So I'm really interested in one, you know, what I've been talking about a lot, characterizing how people respond differently. I think it's fascinating how, you know, I could take a psychedelic and have this like an introspective cognitive journey, whereas another person will take it and they're talking to intergalactic aliens and having some crazy out-of-body experience, right? It's like there's such a range of experience that people can have, and we're not even close to exploring this in psychedelic research, subjectively or in the level of the brain. So I think seeing how these diverse responses relate to the brain and how we can predict them using questionnaire measures, but also even as brain imaging, and what that tells us about different people and how brains differ. I think that's super exciting and relevant for therapy. If we can say, based on this brain scan, you're most likely, and you're 70% more likely to benefit from psychedelic therapy, or you're 50% more likely to need more than 5 sessions rather than 1 to 3. Like to be able to say that on an early stage and have this prognosis based on the brain and based on the brain in combination with other measures, I think that's so important, and brain imaging can help contribute to that. So yeah, and I'm really excited about psychedelics in healthy people. So I really don't think they should be reserved for people with medical conditions only. There's so much more to just being quote unquote healthy or non-pathological. I think psychedelics can help people find greater meaning and purpose and all these things as well. Cool, and my pleasure, everyone.